This topic is covering dihybrid crosses, which are those genetic crosses that you encounter in the higher level questions. And these are when you combine two traits together. So you're examining two traits together at the same time. So students often ask, what's the best way to learn these? Well, in my experience, the only real way to master this topic is to practice lots of exam questions. But the key to that is checking that you're doing them correctly by looking at the answers. So that's really important. But before you can even begin to do these questions, you have to start at the beginning and you have to know what heterozygous, homozygous, recessive, dominant, linked and unlinked all mean, and they're all in the previous videos. So let's try a question. So this is one I've made up. In pea plants, tall is dominant to short. So tall is dominant, so tall is going to get a capital T and small t is going to mean short. And green pod is dominant to yellow, so big G is going to mean green and little g is going to mean yellow. A plant that is heterozygous for both traits, so heterozygous means it has two different alleles for both traits, so this plant is going to have a big T and a small t and a big G and a small g. So a plant that is heterozygous for both traits is crossed with a plant that is short and yellow. Well, short and yellow are the two recessive traits, and so this organism would have to have two small t's and two small g's to be short and yellow. Question 2 says, give the genotype, so the genetic makeup, the alleles, and the phenotype, how they will physically look of the first generation offspring, so of the first generation of children that could result from this cross. I think the easiest way to start is to always start with the letters. Write down what the letters mean, what trait they represent. Then get the genotype of both parents, so write down the alleles that both parents have. So this parent was heterozygous for both traits, so had to have two different T's and two different G's, so two different alleles for both traits. The other plant, he was short and he had yellow pods, and the only way he could be short with yellow pods, because both traits are recessive, is to have two copies of the recessive alleles for each trait, so little t, little g, little g. So now once you've got the genotype of the parents, now you have to do the gametes, the genotype of the gametes that each parent could contribute. So the homozygous parent, the parent that has the two same alleles for both traits, that's easy. They can only give a little t with a little g, so that one's done. But the other parent, the heterozygous parent, can give either t and either g, and an easy way of finding the gametes is to use the word foil. So let's get the gametes from this parent using the letters in the word foil. So this is how you use it. F is the first two different alleles, so that's the first T and the first G. O is the outside two different alleles, so it's the capital T with the small g because they're on the outside. I is the inside two different alleles, so that's the small t with the big G. And lastly, we have the last two different alleles, so that's small t with small g. So we've written the genotype of both parents, and we've also successfully determined the genotype of all the possible gametes that each parent could contribute. So now it's on to do the cross. So we put the gametes from the one parent along the top and the other gametes from the other parent along the side, and we're using a box or a grid called a Punnett square. So all you do is combine the two T's and then combine the two G's, and if they're capitals, write them first. So the first offspring is capital T, small t, capital G, small g, so that's its genotype, and its phenotype is that it's going to be tall with green pods. The next one is capital T, small t, little g, so that's the genotype, and this offspring is going to be tall with yellow pods. The third one is two small t's and a capital G and a small g, so this plant is going to be short and it's going to have green pods. And finally, two small t's, two small g's, this plant is going to be short with yellow pods. So what would happen if you had two heterozygous parents? Well, you'd just use foil twice and you'd have 4 gametes on the top and 4 along the side, and you'd have a larger Punnett square. So this is the protocol we follow. Match the letters with a trait, so make a key. What letter is going to mean what trait? Then write down the genotype of the parents. Then find out what gametes they could produce. So you're going to get the genotype of the gametes. Next, you're going to make a Punnett square, and you're going to put the gametes from one parent along the top and one along the side. And a handy hint is, as you go box by box, write in the phenotype because you will get confused. The questions are usually very similar, so if you practice them, you will get very good at these crosses, and it's a nice question to do. Best of luck.